Hello everyone, my name is Michał Sułek and I am a PhD student at Department of Immunobiology at Maria Curie Skłodowska University. In my presentation, I would like to tell you about the immune priming phenomenon, which is also my main scientific interest, which is also strictly connected to my PhD research. Before I tell you about the immune priming, I would like to tell you about the Red Queen hypothesis by late Van Vallen. Van Vallen took his hypothesis from Lewis Carroll's book Through the Looking Glass and What Alice Found There. And this book is a continuation of famous Alice Adventures in Wonderland. In the book, Queen with Alice ran very fast. However, when Alice stops, exhausted, she noticed that they are still in the same place. Then the Red Queen explains to Alice that here you can see it takes all the running you can do to keep in the same place. This particular sentence was used by a scientist to explain that all species have to adapt constantly to changing conditions in order to avoid extinction. In the context of immunology, it means that both hosts and their pathogens need to improve their weapons. In case of hosts, they need to improve their defense mechanisms, and in case of pathogens, they need to improve their virulence factors. In the 70s and 80s of the last century, the first reports began to appear about the possibility of some kind of vaccination of insects. This process allowed them to more effectively fight the secondary infection. And the first milestone in immune priming research was the work by Faulhaber and Karp, presented on the left side of the slide, in which the term immunological memory was used to describe adaptive properties of insects' immunity. In the following years, there was, of course, intensification of research related to that phenomena of vaccination of insects, which in 2004 was for the first time called immunological priming, which with a slight change was adopted in the scientific community and now is known as immune priming. The following years of research on the immune priming led to the discovery of various combinations in which the first contact with a pathogen or its component provided protection during subsequent exposure to the same or another pathogen. On this slide, I quote a few of these works, and in case of the first one, the pre-infection of insects with non-pathogenic Escherichia coli provided protection for insects against future infection with entomopathogen. In case of second publication, the protective effect was observed after injection of lipopolysaccharide of pathogen of entomopathogenic bacteria. In the third publication, we can observe the true specific immune priming effect. And by true, I mean the full analog of vertebrates memory. In this case, the priming effect was observed after pre-infection with the same pathogen. The immune priming effect also is not only restricted to just one generation. Current trend among scientists which study this phenomena are now mostly focused on transgenerational priming, in which the protective effect of the first infection is passed to the progeny or even to the future generation of primed animals. To sum up, immune priming is a memory-like phenomenon found in invertebrate taxa of animals. A beneficial effect of priming consists of better secondary immune response to a pathogen observed as higher survival rate of primed individuals in comparison to non-primed. It is important that we talk about animals which rely only on innate immunity mechanisms. What is more, and what makes this phenomena particularly interesting and of course hard to study is fact that there is no known rule if immune priming occurs after certain pre-exposure of animal to a pathogen. Also, we can distinguish three general types of priming: priming within a generation, transstadial priming, also known as transstadial immune activation, and priming across generation, transgenerational immune priming, shortcut TGIP. On this slide, I have presented a general scheme for detecting the presence of immune priming. This method can be successfully adapted to a variety of model organisms and their pathogens. At the stage 1, named pre-infection, one group of insects is infected with low dose of pathogen, or it could be also dead cells of pathogen or just PAMP, so pathogen-associated molecular pattern of that microorganism. And the second group is injected with sterile buffer. After a desired period of time, which is usually the time that insects need to silence down their immune parameters after the first infection, both groups are infected with higher dose of pathogen. And the next step is comparison between survival of both these groups. If the group which was pre-infected has higher survival rate than the group injected at stage 1 with sterile buffer, we can say that immune priming effect occurred. In the very similar way, we can verify the presence of immune priming across generation and also across stages. In case of transstadial priming, we can infect larvae and observe survival of the adults. In case of transgenerational immune priming, we infect the first generation and progeny of these animals. And on the scheme presenting transgenerational immune priming, on the first step there are adult flies, but in fact we can also infect larvae and not just the adult flies. And in fact, this type of experiments, which involves larvae rather than adult animals, is more popular among scientists. On this slide, I would like to tell you about three different scenarios of immune priming. As I mentioned before, this phenomenon is very complex. It can occur in different host pathogen systems. And as I mentioned before, we don't know if there is a general rule that immune priming will happen or not. A few years ago, the Professor Kerr's research group, which is a leader of immune priming research, proposed three different scenarios which may be contribute to better survival after secondary infection. The first scenario, labeled as A, can be described as accumulation of effectors. The immune priming effect is the result of both infection, which ultimately leads to better protective effect. In my opinion, the more interesting scenario is scenario B, which in fact is similar to vaccine effect known in vertebrates immunity. What is important in this scenario B, that the second infection takes place when the immune parameters are silenced after pre-exposure to the pathogen. As a result of previous infection, we observe stronger protection to the second infection. And as I highlighted before, this scenario is very attractive for scientists because it works as full analog of vertebrates memory. And in fact, most of the recent papers about a phenomenon of adaptive immunity of insects refer immune priming as an exact analog of vertebrates' memory. And the last presented scenario, which is scenario C, propose whole different strategy. It suggests that the first infection with lower dose of pathogen triggers different immune response than the infection with a single higher dose. In fact, in nature, usually all these three scenarios are presented and are engaged in a single immune priming effect. What is more, as I mentioned, usually priming effect is caused by at least two of these scenarios in one host pathogen system. So as you can see, the complexity of this phenomenon is high. We don't know the exact mechanism which are responsible for occurrence of immune priming. For example, in some host pathogen systems, immune priming is presented due to accumulation of immune effectors like antimicrobial peptides, stronger enzymatic activity, or intensification of microbial clearance process like phagocytosis or nodulation. And as I mentioned before, every single case of priming needs to be analyzed independently. For example, from my research, in case of priming effect presented after Bacillus thuringiensis infection in Galeria melonella larvae, we observed higher activity of phenol oxidase after secondary exposure. This enzyme is known to play a vital role in insect immunity, but however, after pre-infection and later proper infection with different entomopathogen in this larvae, we observed inhibition of that enzyme in prime groups. So it's really different. Every single case of priming needs to be studied independently. But back to the hypothetical mechanism of immune priming. Currently, the most prominent candidates which may be responsible for occurrence of immune priming are Down syndrome cell adhesion molecule, DSCAM, and specific pathogen recognition receptors, PRRS. Basically, DSCAM are proteins which are capable of creating thousands of different isoforms in insects. The amount of isoforms varies in different species. Also, it is known that these molecules are engaged in immune processes like opsonization and phagocytosis. And what is more, these proteins contain IgG domain, immunoglobulin-like domain, so they are similar to vertebrates' antibodies. However, currently there is lack of studies about potential influence of these molecules in immune priming effect. Another possible candidates are PRRS, specific PRRS, and by specific I mean PRRS which are characteristic for particular species or order. The existence of PRRS specific to certain organisms might explain the selectivity of priming effect. For example, hemoline is a PRRS characteristic for Lepidoptera order. This protein has no direct antimicrobial activity, but is engaged in immunological process as signaling molecule. What is more, hemoline, similar to DSCAM, possess Ig domain. So also it has evolutionary similarity to vertebrates antibody. Another scenario which may contribute to priming effect are epigenetic modification. It is proposed that first infection may result in DNA methylation or demethylation or histone acetylation deacetylation. And this strategy, as well as transfer of signals, mRNAs, and effectors, are proposed mechanisms of transgenerational immune priming. And in fact, thanks to advances in gene sequences and use of NGS, there are many studies which confirms these mechanisms in many cases of TGIP. At the end, I would like to say just a word about my current research. So currently my team works on an ancient project named Bioactive Compounds in the Immune Response of Galleria melonella infected with entomopathogenic bacterium Pseudomonas entomophila, modulation of the defense in immune priming phenomenon. My part in this project is to determine which factors are engaged in immune priming in Galleria melonella Pseudomonas entomophila system. And besides this, I'm also investigating properties of new low molecular polypeptides, which we obtain from the hemolymph of infected larvae. I mostly investigate their antibacterial and antifungal properties. Since my results are not published yet, unfortunately, I'm not allowed to talk much about them. But if anyone is interested, I will update him in the future. Thanks for your attention.